Hi everyone, welcome back. Today we're going to be talking about the mucosal immune system, which is in chapter 12 of your book. And throughout this lecture, I'll be using figures from the textbook so you can refer back. The figure number will be in larger text next to the picture so you can look back and find it easily. So today, in our lecture, we're going to be talking about the mucosa as a barrier tissue. There's also, we'll be talking about the organization of the mucosal immune tissue and how that organization influences function. In particular, we'll talk about IgA antibody as well as the mucosal immune response to infection and control the microbiota. So, as we talked about in the innate immune lecture, barriers are very important for the immune system, and the mucosa acts as the main barrier. So, we'll go through the structure of the immune system and many of the top cells. So these are some areas in the body that are covered by the mucosa. And the mucosa is interesting because it is in contact with the outside world, as well as the microbiome. It is also a very large surface area of the body, so this is a really important immune region or tissue. And as I mentioned, the mucosal tissue is in contact with the outside of the body, and one thing that's very important about that is that it's also in contact with the microbiome, or the populations of bacteria that live opportunistically within our bodies. So we can see different mucosal tissues have different types of bacteria, and these bacteria cluster according to where they're found. And we'll be talking especially about the intestinal mucosa and microbiome in this lecture. Okay, so the structure of the mucosal immune system in the gut is made up of several different regions. So the lamina propria, which you can see here, is the connective tissue layer directly below the epithelial layer. And this, in this layer, we see gut-associated lymphoid tissue, or GALT. Another region that's very important is the Pyrus Patches, which you can see in this blue spot. And in here, the Pyrus Patches actually form visible domes and contain B cells, germinal centers, and T cells. And dendritic cells actually project from the lamina propria and Pyrus Patches into the lumen of the gut, which we'll talk about a little more later. So M cells are a special cell type that are found in the gut. They're actually a type of gut epithelial cell, and they transport antigen across the gut lumen, from the lumen. And in particular, they transport this antigen to dendritic cells within Pyrus Patches. And the dendritic cells then proceed to present this antigen to T cells. And one thing that's interesting about M cells is they actually don't produce any mucus, and this allows them to be more in contact with the microbes in the gut lumen, and they are not blocked by the mucus production. Okay, so here's a little bit more detailed view of the organization of the mucosa. So on the far left, we can see the T cells in the Pyrus Patches and how they are in interaction with dendritic cells, which are shown here in this yellow star. And the dendritic cells will present antigen to T cells. Then activated T cells, as you've talked about in previous lectures, will then drain to the lymph nodes and return to the gut via the bloodstream. And we'll talk a bit more about the T cells in the mucosa in the gut, but these activated T cells can then help control infection. Dendritic cells are particularly important in the mucosal layer, and there are several ways that they can receive antigen. So you can see this in figure 1210. So first of all, macrophages, which you can see here crossing the lumen, will take up antigen transport it across the epithelium, and present it to dendritic cells. Goblet cells within the gut can also take up soluble protein antigens, and dendritic cells in the Pyrus patches can then receive these from the goblet cells. Then other dendritic cells can actually be in contact with the gut lumen and transport antigen across to other dendritic cells on the other side of the epithelium. So in particular, CD11b positive dendritic cells produce IL-23 and IL-10. And this will be important because it leads to the development of Th17 and ILC3s. And IL-10 is a really important cytokine for an anti-inflammatory environment, which we'll talk about later. So dendritic cells also produce retinoic acid, and this helps generate regulatory T cells, which reinforce the anti-inflammatory environment started for the IL-10. So macrophages are also a very important cell type in the mucosa. And in the gut, at least, the macrophages are tissue-resident and non-migratory. However, they are constantly replenished from the blood. So many tissues have tissue-resident macrophages that are derived from the fetal yolk sac or fetal liver. However, in the gut, these macrophages are turned over repeatedly from the blood. And these macrophages will reside within or just under the epithelium. So macrophages in the gut, as they would in other parts of the body, will capture microbes and clear dying cells. And in the gut, they constantly produce IL-10. And they present this antigen to DCs, which migrate to lymph nodes and prime T cells. So these macrophages are important for scavenging and tuning the activity of the already primed T cells as well in the mucosal tissue. And they help form the balance between immune reactivity and immune tolerance. So IgA is a very important antibody, particularly for the mucosa and the gut. And this is because IgA, as you guys talked about a little bit earlier in the course, is pretty unique. So IgA can actually be secreted into the gut lumen, and it can be secreted as a dimer. So here you can see in this diagram we have a dimer of IgA that is connected with a J chain. So the IgA is produced locally by B cells in the lamina propria, and a deficiency is actually fairly common, but typically a deficiency in IgA can be compensated for by IgM, so this is not necessarily very pathogenic. So IgA can also bind to the layer of mucus and bind toxins and microbes there. And it can reside in endosomes as well, so when cells take up particles from the gut lumen, the IgA can be within that endosome. So as I mentioned, IgA in the gut can bind to the surface of the mucus and bind to toxins or microbes, preventing them from getting closer and in contact with the epithelial cells. IgA is also able to bind and neutralize antigens in endosomes, as I mentioned. And interestingly, IgA can actually transport toxins from the lamina propria when it's being secreted. So here, when the IgA is being produced, before it's transferred into the gut lumen, it can actually bind to toxins that are on the lamina propria side of the gut. And then it can help remove these toxins as it's secreted. It will travel with the toxins across back into the lumen of the gut, where they're less likely to do damage. So binding of IgA to M cells also helps with the transport of antigen that can then be presented to dendritic cells. So another large category of cells within the gut are the intraepithelial lymphocytes, or IELs. So lymphocytes in the gut reside either in the lamina propria and Pyrus patches, as we talked about, or within the epithelium. So we can actually see in these pictures, especially in this electron micrograph, that the IELs are embedded within the epithelial cells. So this allows them to interact with many different cell types. And this can actually be a few different types of lymphocytes. So we can have Tregs, which produce IL-10 and help balance the immune response from CD4 effector cells. And IELs can also help clear infected cells. So there are two different types of IELs. The first type is type A, or inducible, and the second type is type B, or natural. So type A, or inducible IELs, are found and recognize normal MHC. So in this figure, we can see a virally infected epithelial cell in the far panel, and this cell will then display a peptide to a CD8 IEL via MHC class 1, just like we've talked about previously. Then the IEL can actually go to that epithelial cell and kill it because it's infected, and this epithelial cell will be replaced. So type B, or natural IELs, are a little different. So these cells do not recognize canonical MHC, but instead will recognize this MIC A and B. So there are a few different types of natural IELs. This includes T cells, as well as ILC2 and ILC3, and inducible NKT cells. So these can still result in cell killing, but it is through different recognition mechanisms.